Today we will study about heterocysts. What are heterocysts? These are the specialized structure found in the member of nostocle except oscillatories and toriaceae and stygonematales. They arise from somewhat enlarged vegetative cell and are characterized by the presence of conspicuous pore at one or both the poles. In terminal heterocyst, the pore is usually present only at one end, whereas in intercalary heterocysts have a pore at both the end. The position of heterocyst is a specific character. They are terminal in gliotrachea, intercalary in nostoc and cystosoma, and rarely lateral as in notochopsis. They may be solitary or in pair. Example in Anabenopsis. Intercalary heterocysts are usually spaced regularly along the filament. The structure of heterocysts are thick wall, pale yellow and barrel-shaped structure. The wall is differentiated into an outer fibrous, a middle homogeneous and an inner thin lamellar layer. It is characterized by the presence of pore either at one or both the poles. The wall layers become thicker in the polar region and the pores are plugged with a refractive material called polar granules. Heterocysts contain dense and homogeneous cytoplasm. They contain mostly carotenoid, hence yellowish in color. In the absence of phycobilin, which is the principal light absorbing pigment in oxygen evolving photosynthetic reaction, and chlorophyll A, there is no carbon dioxide fixation or oxygen production in heterocysts. The photosynthetic lamellae thylakoids are tightly packed with reduced interlamellar space. They are concentrate along the periphery in a complex reticulate manner. The lamellae contain two lipid, glycolipid and acyl lipid, which are not found in vegetative cell. Three types of granule, structural granule, crystalline granules and small granules, are associated with the peripheral lamellae, but they decrease in number and eventually disappear as the heterocysts mature. The mature heterocyst may remain attached to the filament for varying period. Development of heterocysts. Heterocysts develop from a vegetative cell. One or both the daughter cell of recently divided vegetative cell may behave as preheterocyst. A preheterocyst undergoes a number of changes to form heterocyst. It enlarges in size and form a multi-layer envelope outside the cell wall. The wall becomes differentiated into an outer fibrous, a middle homogeneous, and an inner lamellated layer. The outer fibrous layer is uniformly thick, whereas the middle and inner layer are thicker at the polar end. The pores appear at one or both the poles, and though these pores, cytoplasmic continuity is maintained with the adjacent vegetative cell. There is a gradual dissolution of storage granules and breakdown of photosynthetic thylakoids. Sometimes the lamellae may form swollen vesicles and they become concentrated along the periphery and at the pore. In mature heterocysts, the polar pore become plugged with a refractive material also known as the polar nodule, due to which their cytoplasmic connection with the adjacent cell are broken. The frequency of heterocyst production is affected by the following external factor. The blue and the green light inhibit heterocyst development, whereas the white and the red light support it. The concentration of phosphate salt in the medium stimulate heterocyst development, whereas the absence of magnesium and iron Iron in the medium inhibit heterocyst development. The presence of combined nitrogen in the medium inhibit heterocyst development. The formation of heterocysts is inversely related to the amount of nitrogen in the medium. The cellular and nitrogen ratio play an important role in inducing and controlling the differentiation of vegetative cell into heterocysts. Thank you.